Welcome viewers. Today in this session we will be talking about generalized transduction. In previous episodes we have discussed what is transduction, how transduction is helping in evolution of bacteria and how it can be used in recombinant DNA technology. In this episode we will be discussing about generalized transduction. What do we understand by generalized transduction? What are the different steps involved in generalized transduction? What kind of phages brings in generalized transduction? And what are the applications of generalized transduction? We will be also discussing about how generalized transduction is useful in studying the genetics of bacteria. Before we move into all of these sections individually, or we take them individually, we'll first recapitulate the definition of transduction. So, transduction is what? Transduction is a major mechanism of lateral gene transfer occurring in bacteria in which the genetic exchange takes place from one bacterial cell to another by means of a bacteriophage. So we know that the lateral gene transfer which is occurring in bacteria are of three main types. One is transformation, conjugation and transduction. Transformation takes place when a fragment of DNA is released from a dead bacterium and taken up by a recipient cell. Conjugation occurs when two bacterial cells comes in close proximity to each other. Whereas transduction takes place when one bacteriophage takes up genetic material from the donor bacteria and gives it to the recipient cell. So we know that the bacteriophages which attack the bacterial cell are of two main types. Broadly, one are the virulent phages and the temperate phages. Now after they are released from the donor bacterial cell, some of the progeny phages might carry a part of the phage DNA, or instead of the phage DNA that might carry a part of the bacterial chromosome or part of bacterial chromosome along with the phage DNA. In this way, they are able to bring in recombination to the recipient cell which are getting injected by the bacteriophage, or rather getting infected by the recombinant bacteriophage. So, in this way we can see how transduction is taking place in bacterial cell. Now, broadly the transduction can be divided into two main types. One is the generalized transduction and the specialized transduction. Let us see the differences between the generalized and specialized transduction with the help of this diagram. So as we can see, this is the donor bacterial cell of the genotypic composition A+ and B+ in both the cases. Now when a phage is injecting its DNA, so what is happening? The bacterial chromosomal DNA is getting broken into small pieces, as well as the phage DNA is also getting replicated. When the phage progeny are being assembled within the bacterial cell cytoplasm, some of the progeny phages might receive a fragmented copy of the donor chromosome and get packaged. And then they are released as the form of phage lysate. The phage lysate, which consists of transducing phage as well as the normal phages, infects another recipient cell of genotypic composition A-B-. And what is happening? If suppose the recombinant phage is carrying A+ genotype, so if there is an homologous recombination with the A- portion of the recipient cell and thereby bringing in recombination. So the genotypic composition of the recipient cell changes from A- to A+. Whereas in specialized transduction, as we can see, the viral DNA is getting integrated inside the donor chromosome in the form of a prophage. Under adverse conditions, the prophage is getting excised off from the donor chromosome along with a small part of the donor chromosomal DNA. Now this part is getting packaged inside the new viral particles which is released on infection to a recipient cell with A-B- genotype. Again a homologous recombination taking place, but here a part of the viral DNA still remains and get integrated inside the recipient cell chromosomal DNA. So here any part of the DNA can be introduced. Here only a specific segment of the chromosomal DNA is introduced into the recipient cell. So there are two main parts. to the mechanism of generalized transduction. The first is packaging of the donor DNA into phage particles and the stable introduction of the packaged DNA to the recipient cell. So, as we shown in this diagram, we can see that the phage DNA along with the bacterial chromosomal DNA might get packaged within the phages. Now, this phage, which is carrying the part of the bacterial chromosomal DNA, is known as a transducing phage. So, this is the first part of the mechanism. The second part is the transducing phage is injecting the bacterial chromosomal DNA. This chromosomal donor DNA is recombining with the homologous portion of the recipient cell in a double crossing over or crossover, resulting in the integration of the donor chromosomal DNA into the recipient cell, and the recipient cell becomes recombinant and is known as transductant.
And what is the difference between a transducing phage and a transductant?
The difference between a transducing phage is that the phage which are carrying, instead of the normal phage DNA, the part of the bacterial chromosomal DNA of the donor cell is known as transducing phage because they are bringing in transduction. And what is a transductant? The transductant is a recombinant recipient cell which is formed after the homologous recombination of the donor DNA into the recipient cell's chromosome. So, in this way, the transductant is the recombinant recipient cell. Let us move to the steps of generalized transduction. The figure which has been shown here is for the bacteriophage P1, which is known to infect Escherichia coli and thus bring in generalized transduction of Escherichia coli. So, let's look the first step. Now, generally the phage P1, it injects its DNA into the donor cell. The donor cell is of the genotypic composition A plus B plus C plus. Now, if it does not enter the lysogenic state, it goes for the lytic cycle. In the lytic cycle, so what is happening? The chromosomal DNA of the bacteria is getting broken into small fragments. Now, one of these fragments might carry the A plus gene or the B plus gene or the C plus gene. Now, these genes or these bacterial chromosomal part might get packaged in the viral progeny instead of their normal DNA, and hence then they are becoming transducing phage. The phages are released from the lysis of the donor bacterial cell in the form of phage lysate. The phage lysate is infecting another recipient cell, say suppose which is having the genotypic composition of A minus. The transducing phage, say for example in this instance, is carrying the part of the DNA of the donor chromosome which is positive for A genotype. So as a result, what will happen? It will find its homologous portion, and therefore there will be a homologous recombination which is taking place due to a double crossing over, and the A minus will become converted to A plus genotype of the recipient cell. So the nature of the recipient cell is changing from A minus to A plus, or from auxotrophic to prototrophic in nature. The released fragment after the double crossing over is digested by the enzymes. So in this way, we can see in the steps in generalized transduction, what is happening? There is a transductant form and a transducing phage which is bringing in this transduction. Now let us move to the features of bacteriophages which can bring about generalized transduction. The features are that most of the bacteriophage, they package their DNA at sites which are known as their pack sites or cos sites. So these pack or cos sites might also be present on the bacterial chromosomal DNA. May not be exact to the sites present on the phage DNA, but they have sequences similar to the pack sites found on the phage DNA. So as a result, instead of packaging their DNA, the phages by mistake package the bacterial chromosomal DNA at or from these sites. The most common example of generalized transducing phages are P1, P22. P1 is known to infect E. coli, P22 is known to infect Salmonella typhimurium. Now both of them, they are good generalized transducers. Now why they are so? Because both of them, they do not have stringency for packed sites. They can also package DNA from pseudo-packed sites present on the bacterial chromosomal DNA of the donor cell. Now as a result, they by mistake package the DNA not from their own phage DNA but from the bacterial chromosomal DNA. Apart from this, they package the DNA into their heads via a headful mechanism. What is headful mechanism? That is, the genetic material or the replicated genetic material is stuffed inside the head of the virion until it is full, and it might contain more than a single copy of the sequence. So as a result, P1 and P22, since they have less stringency for their own packed sites, are good generalized transducing agents. Apart from this, the other bacteriophages are T4 and bacteriophage lambda, but they can be used for generalized transduction only after certain modifications. Like in T4, we know it degrades the bacterial chromosomal DNA into small fragments, and they can be used as a generalized transducing phage.
T4 being a virulent phage is going to degrade bacterial DNA. Then how is it going to protect its own genomic DNA from degradation?
Now what is happening is the T4 DNA, it protects its own DNA from getting lysed or from getting broken by synthesis of a modified cytosine residue. The modified cytosine residue is glycosylated hydroxymethyl cytosine. As a result, the phage is able to protect its own DNA and degrading the bacterial chromosomal DNA. So when we can use T4 DNA? Only when the T4 DNA enzyme, which is breaking down the bacterial chromosomal DNA, has been inactivated. Then we can use the T4 phage as a generalized transducing phage, and it can package any part of the donor chromosomal DNA and does not have specificity for packed sites. Now let us see what happens when the recipient cell, or rather the donor chromosomal fragment, enters the recipient cell. On entering the recipient cell, it might follow or it might face two fates. One, it might go for abortive transduction or complete transduction.
Ma'am, I have a question. What is abortive transduction?
Well, abortive transduction, as the name suggests, in due course of time, the transduction is getting aborted. So why it is taking place? We will see. Now, as we can see in this figure, the DNA which is coming from the donor chromosomal DNA. Now, this donor chromosomal DNA might face two fates. One, it might become circular and may not become integrated into the recipient cell chromosome. Why it is not getting integrated? Because there are certain enzymatic pathways which are required, and that is not taking place. So in this way, what is happening? In this abortive transduction, the fragment which is coming in, it stays in the form of a non-replicating unintegrated fragment. But the genes which are present on this fragment are capable of expressing themselves. So thus the cell becomes partially diploid or functionally diploid for that particular region of the chromosome. In the other case, as we can see that the complete transduction, the rest, the 10% of the DNA can stay in the linear form. Now out of this linear form, the 2% can get integrated into the recipient cell chromosome. The rest 8% is degraded into small nucleotides or to its corresponding nucleotides, which are recycled during DNA synthesis. So what we can say, which is more stable of amongst abortive and complete? It is the complete which is more stable. Why? Because it is getting integrated and it is stably inherited during the cell division by all the cell lines or by all the progenies of that particular bacterial cell. So it is the complete transduction which is more stable than abortive. another important feature of generalized transduction, that is, how do we measure transduction or the rate of transduction. For studying transduction, or in a typical transduction experiment, the donor cell and the recipient cell, they differ from each other by one or more genetic traits or genetic markers. A genetic marker is what it is concerned about a gene whose location we know on a particular chromosome, and it is associated with a particular phenotypic trait or character. So in this way, the different genetic markers can be used to study the different types of transduction, or what is the rate of transduction. So this is one important feature. Now the second important feature is the co-transduction. What do we understand by co-transduction? Co-transduction means we know that the DNA which is getting packaged within the viral head is capable of containing at least 50 genes of the bacterial system. Now, if we want to map the gene distance or study the gene order, it can be easily done with the help of co-transduction. Now, what is co-transduction? The probability of being both the markers, or suppose there are more than one genetic marker being present in the same transducing particle, so it increases the chances of transduction of both the genetic markers into the same cell or same recipient cell. So this is co-transduction. With the help of co-transduction, we can say whether the genes which are located on the bacterial chromosome are closely linked to each other or not. If they are closely linked to each other, they will be packaged in the same viral fragment, in the same viral progeny, and they will become the same transducing phage which is attacking the same recipient cell. So with the help of co-transduction, it helps us to understand how closely spaced or linked the genes are on the bacterial chromosome. Now this can be further explained better with the help of this figure. So let us see, there are two examples which are given. So we can see, suppose this is with the example we are taking it from E. coli. So the P1 phage, which is known to bring in generalized transduction. So suppose it has infected a donor cell with a genotypic composition of leucine positive and galactose positive. So it is able to grow on a medium that is lacking leucine as well as galactose. So now what will happen? The P1 progeny phage, after it gets released from the donor bacterium, it is attacking a recipient cell which is leucine negative, galactose negative. Now what can happen? If the transducing phage, it contains DNA which is leucine positive, so it will undergo homologous recombination at this point, or if it is galactose positive, it will undergo homologous recombination at the galactose site. But we will never see both leucine positive and galactose positive in the recipient cell. Why? Because leucine and galactose gene on the recipient cell chromosome are far located from each other. So in this way, we can get only single transductant in this case. Whereas, let us take up another example where the P1 phage is attacking a donor bacterium which is leucine positive, galactose positive and biotin positive. So what are the then different cases of transduction here? Now since again here what can happen? One, either galactose only. So we will be getting a single transductant of galactose in the recipient cell, or we will get a biotin single transductant of the recipient cell, or both biotin and galactose. Why? Because this is the fragment which is coming from the donor cell. Now this donor fragment is containing both galactose and biotin gene on it. Why both of them are present in the same fragment? Because both are very closely related, or rather closely linked to each other. Why they are linked to each other? Because they are in close proximity to each other. So thereby the chances of co-transduction of both the genetic markers increases in this case, and we get co-transductants in the third example. So in this way we can see, with the help of co-transduction, we can clearly define the linkage of genes found in the bacterial chromosome. We will now see how generalized transduction is useful in studying the genetics of bacteria. For this, let us see, so there are two main aspects in studying the genetics of bacteria. First is gene order, in which arrangement the genes are arranged on the chromosome, and the second is what are the distances between the genes. So the first, let us take up gene order. Now say, suppose for an example, there is a donor cell which is leucine positive, threonine positive, and it is resistant to the poison sodium azide. The phage, the transducing phage, is carrying the piece of DNA from the donor cell to the recipient cell, which is leucine negative, threonine negative, and is sensitive to the poison sodium azide. Now what can be done? After few transduction has been carried on, now we will go for the phenotypic composition of the progeny colonies, or we can say of the recombinant bacterial colonies. So what do we see? The first experiment which is conducted is, there we are taking the selectable marker as leucine. So those colonies which are leucine positive as well as positive for sodium azide are of 50%. So the 50% of them are resistant to both. The second is the leucine positive and only 2% are threonine positive. So from this analysis, what we can say? We can say that the leucine is located close to sodium azide but not to threonine. So it can be either threonine, then leucine, or sodium azide, since both of them are closely linked, or it can be threonine as sodium azide and leucine. Both the cases can be there. Now how can we confirm the position of then leucine and sodium azide? For this, the second experiment is done, in which they take the selectable marker as threonine. Now when threonine is taken as a selectable marker, then it's seen only 3% of the colonies are resistant to both threonine and leucine. That is, they are threonine positive and leucine, whereas 0% are sodium azide and threonine. So this shows that threonine and sodium azide are located far apart from each other, and the transducing fragment is not able to carry such a long fragment. So there are 0% colonies. Whereas in the third experiment, when leucine and threonine are both taken together, then 0% are sodium azide resistant. Hence the gene order of the three genes are threonine, leucine, and sodium azide. So in this way, we can confer or infer the gene order with the help of co-transduction or with the help of generalized transduction experiments. The second is the gene mapping distances. How can we measure the distance between the two genes? Suppose there is a donor cell which is having the genotypic composition of A positive, B positive, and the recipient cell is A negative, B negative. Now there are three main types of transductants which can be formed. One is A positive B, B positive A, or both A positive and B positive. Then how will we understand that what is the percentage of A positive transductants? It will be total number of single gene transductants by total number of total transductants into 100%. So suppose for A positive, it will be A positive B divided by the total number is A positive B plus A positive B positive into 100%. Similarly, for the B positive, how can we go for B transductants? That it will be A B positive divided by AB positive plus A positive B positive into 100%. So in this way, we can see or map the distances between the two corresponding genes present on the bacterial chromosome. Now let us go for the different applications or uses of generalized transduction for visualizing the gene order and mapping the gene distances. So what are the different applications? So generalized transduction has been a useful tool in recombinant DNA technology for construction of cDNA libraries and genomic libraries. It is a very useful tool for or as a vehicle for vaccine delivery. It has been a very useful tool for carrying DNA or normal copy of DNA to the disease cell in the process or in a process which is known as gene therapy. And the other important fields are in construction of isogenic strains of bacteria, in seeing the complementation test, and in plasmid transduction, as well as in the delivery of transposomes. So these are the different areas in which generalized transduction is playing or has been playing an important role.